Hello everyone, welcome back to Chinmoy's Biology channel. So today we are here with a very important question which is coming in many of your exams. Let's discuss about that. The Drosophila, why it is used as an experimental materials in many of the genetic experiments, and we have also seen that in the principles of inheritance and variation it was used as a experimental material by Thomas Morgan as well. So let us see what are the reasons behind that. So without further delay, let's get started. So let us come to the point that what are the reason behind the usage of Drosophila as a genetic studies and genetic studies. So first of all, it has been extensively used for genetic studies because of the following reasons. So first of all, it is that it is a small insect and occupies a very little space. So for handling a big insect or a big animal for these experiments will need a huge space or a huge lab. So it is a small insect which will occupy a small space, or rather a small laboratory. Now life cycle of Drosophila is very short and it is completed within 2 weeks only. So we can perform the experiments and we'll have to wait for only 2 weeks to see the results. So after the life cycle is completed in the resulting offsprings, then again the next generation F1, F2 and all will be observed very quickly. So within 2 weeks of waiting time. If we are taking other animals, might be their life cycle will be more and we will have to wait for 3, 4, 5 months or a year to see the observe the results. Now next comes the Drosophila culture is maintained at room temperature, and so it can be easily raised. So what is the meaning of maintained at room temperature? So it is actually grown in lab. The procedure is very easy and it is grown in a simple and synthetic medium or material. So it is the culturing of this Drosophila is quite easy, okay, and it produces a large number of offspring and allows sufficient data to be collected. Now Drosophila has only 4 pairs of chromosomes that are easy to handle. So 4 pairs means it is having 3 pairs of autosomes and it is having 1 pair of sex chromosome. Okay, it is having 1 pair of sex chromosome. Now, the Drosophila genome is significantly homologous to that of human genome, which makes the study of certain genes much easier. So, as it is homologous to human genome, it makes the study of mostly all the genes easier. Now they show a XXYY type of sex determination. So in the next slide, I'll just tell you in brief that how the sex determination in Drosophila is done. It is a bit different from that of ours. So that type of sex determination is called XX and YY type of sex determination. Now there is sexual dimorphism is also seen in Drosophila. So this also I'll discuss in the next slide only. You just remember these points. These are very important because these are the reasons behind the use of Drosophila as a genetic material. Now use of low power microscope and many heredity variations can be observed. So when you are using a low power microscope only, you can see many heredity variations as well. So the sexual dimorphism and the type of sex determination in Drosophila I'll tell in the next episode. So stay tuned, don't skip in between. You'll have to watch till then because it is a very important and short topic which will be coming in your exams. Okay, so let's move on to the next one. So this is what about the sex determination of Drosophila we want to know. So in Drosophila, the males and the females, they differ in the number of their X chromosomes relative to the number of sets of autosomes. So how it is? Like X is to A ratio. So if suppose which I am drawing, it is the X chromosome, and this is one set of X chromosome, and here with other color I am drawing 2 sets of autosomes. Okay, so 2 sets of autosome. This is 1X is to 2A. Okay, so that is this is the genotype of a male. Okay, this is a male Drosophila. Then what will be a female when there are 2 X chromosomes? And obviously there is that ratio is to 2 autosomes. Okay, 2 X chromosomes and there are two autosomes like this only. So there are two autosomes like this only, two pairs of autosomes. So this will be 2x is 2, 2a will be male. So this is how the genotype of the male and female sex determination in Drosophila is done. So this you can see, this is the male one and this is the female one. There are certain differences, and those differences we are going to discuss in the next slide. That is the sexual dimorphism. That also I told we will discuss. Now let us discuss that. So what is sexual dimorphism? Sexual dimorphism is actually the difference in the appearance between the males and the females of the same species. So and in shape, in color, in size and structure that are caused by the inheritance of one of the other sexual pattern in the genetic material. So how the variation is happening? Now you can see here, this is the female and this is the male one. So the female is having 7 abdominal segments, whereas the male is having only 5 abdominal segments. So the female is having a sex comb where that is absent in female. Now here the tip is dark tip in male and it is a very light tip in female. Okay, so these are several here and there are differences in the body of a male and a body of a female. So this is the sexual dimorphism. So we wanted to have a quick idea about why Drosophila is used in all the experiments, mostly for genetics. Okay, so we should know that in brief, and we will get these questions in our 12th as well as in NEET and all. You can get that. So to have a brief idea, I just explained it. So there are more important topics which are on the way to come. So stay tuned, stay updated, and don't forget to subscribe to my channel and share it with your friend. Thank you. Thanks a lot for watching. Bye bye.